and welcome to Field Notes on Climate Change, reporting in from the front lines of Arctic research. I'm Emma Brisden, and this podcast has been produced in partnership with the Climate Impacts Research Centre and is based at the Abisko Scientific Research Station in northern Sweden. This week, I'm joining Steph Owens from the University of San Francisco as she conducts her research on some of the smallest but most important creatures that inhabit our lakes, Copepods. So today I'm back out in Stordalen Mire, which I've also visited before for an episode on methane emissions coming from lakes. But this time I'm joined by Steph, and we're looking at some of the microscopic life in these ponds. But first things first, Steph, this is a climate change podcast. So with climate change kind of incorporating loads of really different small fields of research, What does climate research actually look like to you?
To me, I'd say I'm really interested in food web dynamics. So like you mentioned, I study zooplankton, and zooplankton are this principal link between the base of the food web, like phytoplankton, and then larger or higher level trophic species like fish, and then even above that. So I'm interested in studying zooplankton dynamics, so things like growth, reproduction, abundance, to kind of get a better idea of what's going on in the system, and then if we can monitor that, see how that's changing in our changing environment.
Great, and you've mentioned a few words there that I was going to pick you up on. First of all, yes, you're right, we're looking at zooplankton and we're looking at copepods specifically.
Yes.
But, before we get started, what are they?
Good question. So copepods are the most abundant group of zooplankton. So zooplankton are, besides being very adorable creatures, they're really, really tiny microscopic crustaceans that live pretty much any aquatic environment, lakes, streams, the ocean, ponds, groundwater even. And they're also one of the most abundant animals on the planet. So yeah, copepods just make up the most abundant group of zooplankton. Yeah.
Okay, great. And one of the things we're looking at in your experiments today is their growth rates. Mm-hmm. So why are we interested in that?
So copepod growth production is the key rate process of secondary production, and so things like larval fish and even later stages of fish, they pretty much at some stage in their life always feed on copepods. So if we can study how fast they're growing, we can get a better idea of their secondary productivity, which is kind of like a snapshot of what's going on rather than abundance, and so that can tell us things that ultimately relate to if they're growing faster, they're going to be reproducing faster, higher abundances, more food available to fish is ultimately kind of the end goal to understand that, yeah.
Yeah, so they're pretty tiny but pretty important. They do kind of underpin the whole food web.
Exactly, so there's the link between the base, which is really tiny, like phytoplankton or even microzooplankton, to fish. So they provide that link that without them, the system, I think, wouldn't really work as well.
And your work up here is based in Abisko this summer. What makes the Arctic a really interesting place to be looking at zooplankton? Because as you said, they occur everywhere, don't they?
Right, exactly. So the Arctic is, is a really changing environment. And so the impacts that we're going to be seeing are going to be heightened in the Arctic. And so looking at things like increased runoff into the systems, we're trying to see if that increased runoff, which is on one hand introducing nutrients, but on the other hand limiting light, which both affect the base of the food web, so things like phytoplankton.
Runoff of what? Of organic matter and kind of plant residue?
Yeah, exactly. So when runoff coming into the system, it's picking up organic matter from the surrounding terrestrial environment coming into the system, and how that is impacting secondary producers, so zooplankton. So like I said, on one hand, limiting light, which could decrease productivity, but also, on the other hand, introducing nutrients, which could potentially increase productivity. So we want to be able to better understand, measuring across a gradient of organic carbon that's occurring here, how their growth rates relate to that, to see what we can expect to see if we do see increased organic carbon into the system, how we can expect secondary productivity to be affected.
So you're measuring several different lakes out here, aren't you, in the productivity? So of each of these different lakes, are you expecting to see a difference in dissolved organic carbon? So then maybe that would indicate a difference in, or result in a difference in copepod growth?
Exactly, yeah. We're trying to get, so we have 3 sites. We're trying to get a gradient of dissolved organic carbon. And they're very different sites. So one of them is a really small pond, the other one's a much larger lake, and then another one is kind of in between, has fish, very different surrounding environment. And we want to see that gradient so we can more pinpoint how the DOC, dissolved organic carbon, is affecting growth rates.
And which lake are we at today?
We are at S2, which is the kind of in-between site that I mentioned. So there's actually fish at this site, grayling, which is within the salmon family. And it's a medium-sized type lake.
Medium lake. Great. So run me through your whole experiment. What are we actually doing?
So we're setting up growth rate experiments. So to do that, we're going to first collect some plankton with our plankton net, then get a very narrow size of copepods. So that just represents copepods that are within the same stage. So that means they're in the same life cycle. So maybe think of it as like a class of second graders.
Okay.
And then we're going to be measuring how big those copepods are at that initial time period when they're collected. And then we're also going to be coming out 2 days later, or 24 hours later, and then also 48 hours later to measure how big that group that started out at the same size, how big they are 1 and 2 days later.
Okay.
So I guess think of that maybe as coming back to see a group of 5th graders and how tall they are or something.
Right.
And so if we know how big the copepods were at the initial period and then how big they are at the final period, we can measure that change in mass over time to see how fast they're growing. Yeah.
Okay, great. Well, I guess we better go and do some field work then.
Yes.
Well, now we've dragged this kind of slightly small cone-shaped net with us. I'm guessing this is to catch some zooplankton.
Yeah, this is a zooplankton net. And so it has a certain filter size or mesh size. So this is 100 microns. So we're going to tow it through the surface of the water, and then anything that's larger than 100 microns that we pick up will get kind of pushed towards the back and kind of stay in this little cone jar type thing that we can empty and collect the sample from.
Okay, great. Is there a proper technique, or just kind of swish it around?
Try and try and keep the top of the net just like kind of right below the surface, and then kind of go kind of slow enough that you're going, that you're not like disturbing the water too much, but fast enough that the water you're in there isn't gonna spill out.
Okay.
Yeah, you'll be a champ.
Yeah, you're saying you. Does that mean I'm giving it a go?
Yeah.
Oh,
it's super easy. You'll be great. Yeah.
Right. Okay, so we funnel all of that into a little. Oh, good God, that is.
Adorable, right?
Oh, that's completely. I don't want to say gross because that makes me sound very unprofessional. Oh, it's right. Okay, we've got this tiny little jar, and it is absolutely ram-packed full of these tiny little. I mean, they're bigger than I expected.
Yeah.
I thought they would be. When you said you were looking at them under a microscope, I imagined you wouldn't really be able to see them.
Yeah, they're about the size of like a grain of rice, I'd say, maybe like a little bit smaller, half a grain of rice.
Yeah,
maybe.
like the end of a grain of rice.
Yeah.
But they're just flitting around all over the place.
Yeah, they're jumping around.
My whole summer in Sweden, I've been drinking from the streams here, and I know this is a lake, but this has really put me off doing that.
Yeah. Well, this is super concentrated, but yeah, our chances are probably pretty well that you've consumed a copepod in your life.
Lovely. I mean, it's all natural
just protein.
I suppose. Yeah. Why do you love these guys so much?
I know, right? Well, they're super cool and adorable. You can see them jumping around.
Yeah, they're actually very mobile.
I don't know if you can see like their antenna.
Hang on. Get up close.
They're it's almost like their body lengths.
Yeah.
Yeah, and so they actually kind of use those like radars, and they can sense vibrations in the water to detect prey and also kind of detect predators. And they can also use those antennae to propel themselves forward. And they're also the fastest animal on the planet.
Really?
Because they can go up to 500 times their body length in 1 second.
Oh my goodness.
Yeah. So not only are they most abundant, they're also the fastest. So everyone should love copepods.
Pretty incredible. Okay, I'll give them that.
Yeah.
And I can kind of see I would say there's a couple of different species in there, aren't there? There's some kind of really, really dark brown ones, some kind of red ones, and some almost clear ones.
Yeah. So we have.
Which ones are we interested in?
We're interested in the darker, kind of longer bodied ones. So those are calanoid copepods, and they're often thought to be kind of like the best food for fish. They're kind of juicier and more nutritious, rather than cyclopoid copepods, which are a lot smaller and harder to detect for fish. And then also those clear things you're seeing are Daphnia, which is a type of zooplankton, not a copepod.
Oh, okay.
But they're kind of cool. Not as cool as copepods, but they're still cool.
We get it. You like copepods. Okay, I could maybe be converted. They are quite cool.
Yeah, and some of them you can see have egg sacs, so they're actually carrying little eggs attached to the bottom of their bodies.
Oh my goodness.
Yeah, so those are the adult females.
And they do look quite incredible just in this little jar. They must be pretty amazing under a microscope.
Yeah, I think that's when you can really, truly appreciate them under the microscope.
Okay. Okay, cool. So we've collected them. But what you've just made me do now was all great fun for me and very exciting.
Yes.
But that wasn't really what you're doing today, because you did this 2 days ago, didn't you? What we're actually doing is looking at what's happened since.
Exactly. Yeah, so 2 days ago I came out here, collected zooplankton, kind of put them through different filter sizes to get a very small cohort of copepods of the same size and stage, so early Copepodites. And then
can you just tell by looking at them now?
Yeah, now I'm a pro now. No, I just kind of hold them up and count, and I can tell based on the size like what kind of stage generally they are. And so once I get the size range that I want, I'll take a sample of that, preserve it, so that's going to represent our initial sample.
When you say preserve?
Kill.
Yeah.
2% glutaraldehyde. Yeah, we want to try and get a snapshot of the size that they are. So, for the sake of science, yes, they must be sacrificed. But they are the most abundant animal on the planet. So.
Okay. That's how we're justifying it.
Okay. Yeah. Yes.
So we've got a kind of representative size of what they look like when you first collected them.
Exactly. And then from there we preserved some, but we're gonna let some from that same original size continue to grow in the field in these little bottles. So they're staying in the field in the same temperatures, and they have filtered site water that allows them to be exposed to things that they would naturally be exposed to in the water, but things that aren't bigger than them that could potentially consume them. So then 2 days later, today, we're coming out here and we're going to filter them down, again preserve them, and these samples will represent our 2 days post incubation setup to represent the final size during that incubation period.
Okay, our 48-hour growth time.
Yeah. Okay,
so that's why we're kind of looking at the lake right now. You've used armbands and flotation devices to keep them afloat, which is super nifty. It means you can't lose them as well because they're bright orange.
Yes, exactly.
But we've got these suspended tubs, so those are your incubation chambers from 2 days ago, right?
Exactly.
Great.
Yeah, so now we're going to take the bottles out and filter them down over on a sieve that is going to collect anything larger than 53 microns. So we'll get the copepods that we're trying to get. So just filtering them over the sieve.
Oh yeah, you can see them getting caught.
Yes, so anything jumping around on the sieve, that's what's going to be filtered into this little 20 milliliter vial. So
how do we get them from the sieve?
So what I'm doing now is just using a squirt bottle and kind of rinsing the sides of the sieve with a funnel and pouring it into a little vial.
Oh, there we go.
And so now you can see them kind of jumping around in there.
Yep, definitely still moving.
Yeah, so that's a good thing.
Not for long.
Unfortunately, their time is limited. So now we're going to preserve them with the 2% glutaraldehyde.
Okay.
To kind of get that snapshot of their size that they are at this instant.
Great.
Yeah.
Okay, and then it's back to the lab with them.
Exactly, yeah.
Brilliant. Okay, we're all Copepodded out. We've collected all of our samples for today. Quite a short field day, actually. It's quite nice for me.
Yeah.
Anyway, so what happens next? We've still got time left in the day, so we're going back to the lab. What are we going to do with these guys?
Yeah, so they are preserved now. We're going to be taking them back to the lab, looking at them underneath the microscope, selecting for the species that we want, which is Eudaptimus graciliotis.
Try saying that twice.
I've had to practice a few times. And so once we've selected for the species that we want, kind of organize them so that they're not touching or overlapping. And then using a camera that's attached to the microscope, we'll take a picture of them.
Oh, cool.
And then using a software called ImageJ, we'll actually convert those pictures of the copepods into silhouettes and then ellipse, which is just kind of the circular shape that the copepod ends up making. And from that, we can use the volume equation for an ellipsoid to get the volume of the copepod. And from there, we'll get a ton of data for each individual copepod's volume. And we can take the average for that groups and see the change in volume over time and ultimately convert that to carbon.
Great, and you've actually worked on similar projects before, haven't you, in the northern San Francisco estuary? How does this compare to that?
Yeah, so this research is actually based off of my master's research that I did in San Francisco. And very similarly, I was measuring copepod growth rates on a different species, Pseudodaptimus forbizae. I had time to practice that one.
Yeah, you did that very well. I'm not even going to try. So.
Also a very adorable calanoid copepod.
I'm not with you on this adorable thing. I think I like things that are slightly bigger.
I think you have to see them under the microscope, and then you'll truly appreciate them.
Okay.
Yeah. So I was studying how fast they were growing in the northern San Francisco estuary, and they're a major concern there because they're the principal food for delta smelt, which is an endangered fish in California and an indicator species. And so there's a lot of regulations as well as, just general public concern about the fish's status. And so there's a lot of management actions involved in trying to boost the zooplankton productivity. And one of the methods being looked at is managed flow pulses. So sending fresh water through the system to export copepods from an area of high productivity to an area of low productivity. And so the area I was working in, the northern San Francisco estuary, after measuring their growth rates, I could firmly say that they're more productive there. And so it'll help management decisions about whether, yes, if we could potentially get some more fresh water through the system, we can push more food further downstream to where these delta smelt are.
Great, so that's kind of like a conservation perspective for these endangered species.
Yeah.
Do you think as well, we're facing a future where our fisheries and our oceans are not drying up, that's the wrong word, but we're overfishing quite heavily, So there's a lot of money going into creating fish farms and sustainable fishing. Do you think that, your copepod research could possibly be used for boosting the productivity of commercial fish farms as well?
That's a good question. I mean, I think when you're dealing with the ocean, it's just such a bigger landscape that it's hard to kind of scale up, and it's a little bit easier with like ponds or
estuaries.
Yeah, where it's kind of a bit more confined, but because you have to remember they can move, they're changing with the water, and so I think it's hard to say, but I think it definitely, like, if you could measure their productivity under certain conditions, you could see how those conditions, if they're going to change. If you can get a gradient of those conditions, you can measure the conditions rather than the growth rates, which is actually pretty labor intensive, to get a better idea of what kind of productivity you could expect.
Yeah. Okay, well, we're all done with our fieldwork today. We've reconvened back at the research station in a slightly echoey, kind of rumbly lab, so I don't know how the audio quality of this is going to be, but we're stood in front of a pretty badass looking microscope, just with like a camera attached to a tube at the top.
Yeah, it's literally just a digital camera attached through this kind of adapter type pieces that make it so that you can see what the microscope is seeing. And from there we can take a picture of what's underneath the microscope.
So this is where you said to me that I was going to possibly fall in love with copepods as well.
Yeah, we'll see.
So, okay.
Take a look.
They look pretty tiny on the plate now, but then if I have a look in...
Yeah.
Oh, okay.
So you can even scale...
Oh, wow.
But you can scale to different magnifications, and you can see they're a lot more detailed now.
Yeah, they really are. They're actually... I kind of get it. They're actually quite cool. So they've got these two long, antenna almost, kind of floating arms, and then like almost like a fan at the end.
Yeah, so they have about 5 pairs of swimming legs, and then in the back they have also kind of parts that stick out a lot shorter, but also help to swim.
And we can see the eggs that you were talking about as well. When we saw them when they were live and they're in the pot, you can actually see the eggs now attached.
Yeah.
Wow.
So they keep the eggs attached right to the bottom of their bodies. The adult females do.
Yeah.
And then when they're ready to hatch, they'll break off and hatch into nuclei, which are essentially baby copepods. And they don't look really anything like that, but they'll go through 11 molts or stages until they're ultimately into that final adult copepod.
Oh, wow.
Yeah.
Okay, well, you've won me round. They are actually quite cool when you can see them properly and not just little dots floating in the water. And this seems to be as good a time as any to leave Steph to it, as she has rather a lot of individual copepods now to photograph and to process. And I've got a Skype booked in with researcher Danny Lau from Umea University, who is supervising Steph's project here in Abisko. So Danny, let me start by just asking you the question that I ask every researcher that I try and get on this podcast. What does climate change research actually look like for you and your work?
I'm an aquatic ecologist. So to me, climate change research is to understand the food webs, the biodiversity and functioning of aquatic ecosystems, and how these will be affected or will change with a changing climate. Climate change can affect the aquatic ecosystems alone, but it can also interact with other existing environmental pressures such as eutrophication, acidification, etc. And to me, these interactions may result in synergistic impacts, and these could be more difficult to predict. So I think climate change research is also about investigating the effects of these interactions on the aquatic ecosystems.
Sure. So can you tell me a little bit about your research specifically and maybe where Steph's project fits in with what you're doing?
My research focuses on 3 major aspects: the food webs, biodiversity, and functioning of the aquatic ecosystems, such as lakes and rivers. And I'm also interested in their responses to the environmental alterations. Under the theme of climate change, I ask questions such as how the quantity and, for example, the quantity and nutritional quality of the food resources for the zooplankton will be affected. Or actually, these food resources not only support the zooplankton, but also the whole food webs, for example. And I would like to know what are the consequences for the higher trophic levels, including the zooplankton, benthic invertebrates, and fish. And will they change their diets, or will their growth and production be affected? And Steph's study is to compare the growth of zooplankton in lofton lakes and ponds with a gradient in the concentration of dissolved organic matter. Under climate change, the inputs of dissolved organic matter from the terrestrial environment to the freshwaters are expected to increase. And the dissolved organic matter itself can be a food source for zooplankton, but it can also make the water become darker, limiting the light availability for photosynthesis and for the production of phytoplankton. And in comparison to the dissolved organic matter, the phytoplankton has much higher nutritional quality and is the major food item for zooplankton. So now the question is how the changes in the basal food resources will affect the zooplankton. Here I think Steph's study fits in very well, and her findings will help us to understand whether zooplankton growth will respond positively or negatively when the dissolved organic matter in water increases.
And what do you expect to happen there? Do you expect them to increase or decrease in population size?
Many of the lofton lakes are nutrient poor at the moment, and the phytoplankton production can be low. So in a changing climate, I think the copepod populations in the lofton lakes or ponds may increase initially because of the increases in the supply of the terrestrial dissolved organic matter, which can subsidize zooplankton. But if the dissolved organic matter continues to increase, it will come to the ponds to inhibit the primary production or the phytoplankton production. This means the zooplankton will have less high quality food in the environment. So I guess the long-term consequence is that the nutritional quality and the production of zooplankton will reduce in the future. And this may result in a chain effect in the high trophic levels. For example, the production of fish will decrease as well. This is the food web perspective. But in the biodiversity perspective, for example, the distributions of different organisms will change with climate change. For example, the range of species in the warmer temperate regions may expand to the Arctic, while those more co-adapted species in the Arctic, the range may reduce. So there can be more cases of species invasion and replacement in the Arctic ecosystems in the future. And this may result in changes in the biological communities, trophic interactions, and food web structures. And I think the Copepods cannot avoid these changes, but these impacts are more difficult to predict as this involves multiple organisms at the same time, and their responses to climate change can be species specific.
And do you think there's anything we can kind of do to limit the impact that it might have on these really diverse biological communities?
Of course, we can do something. I mean, together. For example, how we have a lot of efforts in doing the research. I mean, as a scientist, to understand the effects of climate change on the organisms is to increase our knowledge level, for example. It's very important for us to make predictions and to talk to for example, the policymakers to implement the suitable policies to prevent further changes in the long-term ecosystems, for example. But the major cause is climate change. So this involves multiple efforts at different levels as well. So as a scientist, I think our response is to communicate our results to the general public, and also to, on the other side, to negotiate or to convince the governments or the policymakers to implement policies that could limit the changes in these ecosystems.
Now, I quite like Danny's response to that question because it's pretty similar to the response that I've gotten from many of the researchers working on the various facets of climate change here in Abisko this summer. A lot of the research that I've been sharing has felt quite niche, but it all fits into this movement and drive for strengthening our understanding of natural processes in order to understand then how they might be affected by climate change, and also to improve the accuracy of our global climate prediction models. When we're discussing the future of our planet in the face of climate change, and indeed the things that we need to do to mitigate and to combat these changes, we need to come to the table prepared with as much knowledge and as great an understanding as possible to make sure that our plans are as well thought out as they can possibly be. And that's exactly what the scientists up here in Abisko are doing. Thanks for listening. This is the last episode in the series, so I hope you've enjoyed joining Steph and I as we sampled the microscopic communities here in our Arctic lakes. If you head over to Instagram, you can see what our Copepods actually looked like, and who knows, you may even find them as cute as Steph does. You can get in touch and let us know what you think of this episode and of the rest of the series by tweeting the Climate Impacts Research Centre at Arctic Circ, or myself at Emma Brisdian. And don't forget that you can always go back and listen to any of our other 6 episodes. Thanks for listening to Field Notes on Climate Change.